Okay, so today we're going to be talking about chapter 10, which is about muscle tissue. So these are the three types of muscle tissue. The first one is called skeletal muscle tissue, and this is basically the muscle tissue that covers all your bones, and the movement is voluntary. So you can move it on purpose by thinking about it and doing it. And it is multinucleated, so all of these blackish bluish dots here are nuclei, and it is striated. So that means going this way in this direction across each individual muscle cell is a thin and thick filament called actin and myosin, which cause the contraction. But we'll get into that a little bit more later. And then the middle one here is smooth muscle tissue, and it's not striated, and the movement is involuntary. So you can't move it by yourself by thinking about it. It's also not striated, and it covers the walls of your blood vessels and hollow organs. So that's why it's involuntary, because you don't do those things on purpose. You don't move your blood vessels on purpose. It just moves on its own and does its job without you thinking about it. And then cardiac muscle tissue here at the bottom is also involuntary. Its purpose is basically to beat and spread blood throughout your body, and that's why it's involuntary, because you don't think about your heart beating, it just happens on its own. So skeletal muscle tissue, like I said before, covers the bones of your body, and it has stripes called striations, which basically cause the contractions, and you can move it on purpose. Smooth muscle tissue covers the your internal organs, some of them, and your blood vessels, and it's involuntary movement. And then cardiac muscle is involuntary movement, and it beats to spread blood throughout your body, and it's only found in the heart. Okay, so skeletal muscle has 3 layers of connective tissue that cover each part of it. So the muscle as a whole is covered in epimysium, and then a muscle fascicle is covered in paramysium, and a muscle fiber, which is an individual muscle cell, is called endomysium. So this here at the top is one muscle, and that's covered in epimysium. So you can see this lining here on that side, that's called epimysium. And then each one of these individual circle-shaped blobs here, this is one of them, sticking out, those are called muscle fascicles. So this is one muscle fascicle here. This is this same thing as this, except it's zoomed in. So this is covered in paramysium, and a muscle fascicle is made up of individual muscle fibers, which are individual muscle cells, but they just call them fibers. So this is one muscle fiber, this is one muscle fiber, and it's just a closer up image of this. Those are covered in endomysium. So epimysium, then paramysium, then endomysium. This is a histological picture of what I was just talking about. On the outside here, you can see it is covered in epimysium, and then this individual part right here is a muscle fascicle covered in paramysium, and then this individual pinkish reddish part right here is one muscle cell or one muscle fiber covered in endomysium. So this is a muscle fiber. On the outside, it has a plasma membrane. They call it a sarcolemma because it's a muscle cell, and muscle cells are a little bit unique in comparison to regular cells. And then a sarcoplasm, which is a cytoplasm, but they call it a sarcoplasm because it is it's for a muscle cell. And then you can see here on the side, and you can see here on this individual myofibril, which make up the muscle cells, you have these darker regions and these lighter regions. The darker regions are called myosin filament, and the lighter regions are called actin filament. And those are the filaments that cause a contraction in the muscle. So here is one myofibril out of the muscle cell. Here it is closer up. You can see this purple part right here is the myosin filament, and the green part is the actin filament. So this zigzag line here is called the Z-disc, and it's made of actin filament. Same thing with this one. The distance from one to another is called a sarcomere, and everything in between is included in that. So these filaments are separate from each other, but they're both very involved in the contraction of a muscle. Another thing you can see here is this yellowish structure called the sarcoplasmic reticulum, which is the same thing as an endoplasmic reticulum, but they call it a sarcoplasmic reticulum because it is a muscle cell. And basically its purpose is to store, release, and retrieve calcium during muscle contraction. So I'll explain that in a little bit. Here is another histological picture of a muscle or skeletal muscle cell. There's two of them here, this one, and they lengthen in this direction. But the thin and thick filaments actually go in the opposite direction, and that's what causes the striations or stripes. So this, going back to the sarcomere, from one Z line to another Z line or Z disc, either way, the thin filament is the green, the thick filament is the purple. These little bumps here coming off of the thick filament will attach to these binding sites on the green thin filament and pull it closer. So when muscle, that happens during contraction. So when the muscle contracts, the sarcomere gets shorter. So here you can see the thick filament, this purple part. This is what it looks like closer up. These heads here coming off, they stick up. Here's a close-up image of one single myosin head. Has these things on it called actin binding sites, and then it also has ATP binding sites for ATP, which is energy. So when it contracts, it will attach to the actin, or the thin filament, and it will pull it closer to itself, and this head will bend backwards at the flexible hinge region. So it won't look like this, it'll look like it'll going in that direction. And then the actin here, which is the green one up here, this is what it looks like closer up, has these darker green circles right here called the myosin binding sites, or the binding site for the myosin. But they're actually covered in this yellow band and these yellow circles called tropomyosin and troponin. And they need to be moved out of the way in order for the myosin head and the myosin binding site to attach to each other and cause a contraction. So basically how that starts is at a neuromuscular junction. A neuron with an action potential, which is an electrical stimulus, will come down the axon to the axon terminals, and they will hit the sarcolemma of a muscle cell, an individual muscle cell. You can see here, this is an axon terminal and the sarcolemma of a muscle cell. And an action potential will basically release the neurotransmitter from an axon called acetylcholine, or ACH. And they'll come out in these little vesicles here. You can see it in the bottom. And when they come out, they will attach two at a time to these purple channels. You can see this one's closed and this one's open. When the ACH attaches to the channels, it opens them. And when they're open, it allows sodium ions to flood into the cell. And when that happens, it will transfer this action potential from the neuron to the muscle cell. And when that happens, it will eventually lead to the contraction of the cell, but it has to start from the action potential from the neuron, which causes eventually the sodium to flood into the cell. And when that happens, the action potential, here's the sarcolemma again, will go through these little holes in the sarcolemma into these things called T-tubules. And the T-tubules are attached to the sarcoplasmic reticulum that I mentioned earlier, which is the same thing as a smooth endoplasmic reticulum. And then they will release the calcium that they have stored in them into the cell. And when the calcium is released, it attaches to the troponin and moves the troponin and tropomyosin out of the way so that these myosin binding sites are exposed. When the myosin binding sites are exposed, the myosin head with the actin binding sites are able to attach to them and pull them closer if there's ATP available. So when they pull them closer, this flexible hinge region will pull it closer. You can see here in this one, it's straight, and then it binds to the site and bends itself. It'll pull them closer, the whole muscle will get shorter, and only the dark myosin filament will be visible when that happens. And it will hydrolyze the ATP into ADP and PI, and it will release it, and then it will detach itself from the actin filament. And then when the ADP and PI turn back into ATP from somewhere else in the cell, they can come back and then the whole thing could happen again. And it basically looks like the actin and myosin are sliding against each other every time the myosin attaches and pulls the actin closer to itself. So this is kind of what it looks like. The actin or the myosin heads will attach to the actin filament, pull itself closer. And this is what it looks like. This section here will disappear and the muscle will get shorter, because you can see here these myosin heads are attaching to the actin filament. When the tropomyosin is moved and ATP is available, and it will make the muscle shorter. Okay, moving on to cardiac muscle. Cardiac muscle also has actin and myosin filaments, but and they are striated, but the striations come from something called intercalated discs. They're also an involuntary movement, so it's a bit different from skeletal muscle. So these intercalated discs, you can see them here. Here's what they look like in a micrograph. They're actually just spaces in between individual cardiac muscle cells. And they have, here's what they look like when there's a space in between the muscles, or the cells. They have these things called gap junctions, which are these green things. So when there's a calcium release in one of these cells, which causes the thin and thick filaments here to contract, the calcium will leak through these gap junctions into another cell, and it will basically cause a contraction. And the contraction will keep going throughout the cardiac muscle cells. And that's basically how cardiac muscle works, to contract. And then smooth muscle is very different. It does not have striations in the same way that the other muscle cells do. It does have actin and myosin, but it's very different, and the contraction is involuntary, and they're not striated, as you can see here. And that's the end.